Okay, now on to the next one. We're going to talk about a lot of stuff. So we're going to talk about muscle spindles. We're going to talk about Golgi tendon organs. We're going to talk about the chemoreception and how you get, like how your body recognizes smells, tastes, specifically salty and sour, and then sweet, bitter, and umami, because those have two different pathways. And then we're going to talk about sound, what the inside of the ear looks like, and then we'll talk about how your ear can convert those pressure waves from air, which then are transferred to pressure waves in fluid, which then how that transfers to action potentials that your brain understands.
I see.
Great. We're going to start with muscle spindles. We're going to use red because that's a super exciting one. So imagine this is your bicep. Super big, super strong.
Of course.
Yeah. I did totally just do that in the video, so it's fine. Inside your muscle bellies, you have little areas called muscle spindles. Oh, that's weird. It's got like blue on it. So inside muscle spindles, you have two separate types of stuff.
I like stuff.
So this one, that's called a chain fiber. And that one is called a bag fiber.
Ah.
Bag because it makes a little bag.
It has bag.
Yes. And so all along each of these things are tons of mechanically gated stretch receptors. So if they detect any stretch, they open. And so running along these things. So on this one, this is a sensory neuron, and it is a sensory 2 neuron, if I remember correctly.
What does that mean?
I'm going to explain.
Okay.
Okay, and then there's this one.
Ooh.
And this is a sensory 1A neuron.
Oh.
So, but they're both sensory neurons, they're both myelinated, both super quick.
Okay.
So what these do is these are super sensitive to any kind of stretching. So if you stretch, say you're like, super tired and devotional, and your head starts to lean forward, and then you immediately pick your head back up.
Why would I ever be tired and devotional? But continue.
So what happens is these neurons detect stretch, and so they send information to your spinal cord, which I'm going to draw super, super amazingly. There's your spinal cord.
How anatomically correct of you.
It's not, but anyway. So these go into your spinal cord. Actually, I'm just going to join them up for ease of making you understand. Just know that there are two different types of fibers within there. One
2
of them,
and 1A.
Yeah, so the bag fiber detects stretch as you're moving, and this one detects stretch when you're either in like a resting state, so when you're no longer moving. So like here or here, so this is your chain fiber, now you're using the bag fiber, and then chain fiber.
Okay.
Okay, so I'm going to erase that. I need an eraser. Haha, here it is. Just so things make sense. I'm going to draw an anatomically correct arm.
Awesome.
There's your arm. You have muscle here. There's your biceps. And then here's this one. That's your triceps.
Right.
Biceps. Triceps. Okay. And we will. I'm going to erase this one. have that. And we're just going to have our single sensory neuron coming from there. We also have sensory neurons here, but I'm not going to worry about that quite yet.
Triceps are unimportant.
No, they're very important. I literally just use them to do this.
Ah, right.
Okay, so what this sensory neuron does is it goes in here, and to each muscle there is what's called an alpha motor neuron. And so that does the main contraction. So it goes to all of those different little muscle fibers, causes them to contract.
Okay.
And that comes from, information from the brain.
Correct.
So this is our alpha motor neuron.
Okay.
So what this sensory one does is if it's detecting stretch when it's like, hey, we're stretching, this isn't great, it sends information to the alpha motor neuron to say, hey, we're stretching, contract. And so this is an EPSP, or excitatory. Causes this one to actually do something.
I see.
What it also does is it releases, same thing, releases an EPSP onto a funky little interneuron, super little tiny thing, which then releases an IPSP, or an inhibitory, onto the alpha motor neuron that goes to the antagonistic muscle. So your biceps will pull your arm in, but your triceps extend. See you later. And so what that does is when we have the sensory neuron that's detecting stretch here, it will cause the biceps to contract and cause the triceps to extend. Hence, like with your neck, this will, as you're slowly falling down, causes this to contract and these ones to relax, so you don't keep your head down or rip your bones. And so that's the muscle spindles. I'm now going to draw on the Golgi tendon organs. What Golgi tendons do is, so they're little neurons that sit in here, and so they detect stretch, and then you're going to have a little interneuron that's going to go there. And so these detect stretch within the tendon itself. So say you're trying to, curl a super heavy weight, but it's too heavy. So your body is feeling a lot of tension here, but there's no movement happening. So what happens is this activates your Golgi tendon organ. And so this says, hey, we're going to break something. It's kind of like a fail-safe for your body so your muscles don't rip your bones apart. So over here, I'm going to use red. Yeah, I'm going to use red now. So it sends EPSPs. So it'll send an EPSP here to this little interneuron, which will then send an IPSP to the muscle that is trying to contract but can't. But if you notice, I have the second branch and it sends an EPSP to the alpha motor neuron of the antagonistic muscle. So the Golgi tendon organ sends message to this interneuron, which causes the motor neuron for the muscle that's getting stretched, contracting but not moving, to release. And it also causes the antagonistic muscle to contract. So if you're right here and you're trying to pull in really, really, really hard, but you can't, your body, eventually you'll just give out and you go, ah. And so that's your Golgi tendon organ saying, stop contracting, cause the other one to contract so we don't rip our bones apart. And so that's muscle spindles and Golgi tendon organs. Okay, now I'm going to talk about smell. I'm going to draw a very anatomically correct nose. There's our nose. There's a, goes down and stuff. Cool. So anyway, so we have this area right here. Okay, still good. And let's use green. This is our olfactory bulb. This is a bundle of nerves that ends up going to the brain. This right here is a piece of bone. This is called the cribriform plate. It's perforated, and because of that perforation, we have neurons that sit in here, and they can extend. They extend their dendrites into that little mucus area. So what happens is, say we have some fun smell. You're smelling your dog's poop. Hip hip hooray. So that smell is made up of a bunch of different little chemicals, and so those chemicals will go in and bind to that sensory, like, hair neuron. So I'm going to blow that neuron up. So here's our neuron. Here's our little ligand gated channel. Okay. So what happens? That ligand binds right here, and it connects to a G protein complex. So we've got, here's our nose, cribriform plate, olfactory bulb, ligand binds to this. So this G protein activates adenylyl cyclase, which converts ATP into cyclic. AMP converts AMP into cyclic AMP. And so what that causes is we have channel here. When we have cyclic AMP bind to this, we have a rush of calcium and sodium, which will then depolarize this special, like, I don't remember the name. It'll depolarize these sensory neurons. So then what happens, we're going to use green. When the cell depolarizes, we've got our pluses, minuses outside. This opens a voltage gated chlorine channel. And so when the chlorine leaves, this depolarizes the cell even more, causing it to hit threshold. And so it then sends action potentials through here, through the olfactory bulb, and then to the brain, and says, hey, this is what we're smelling. And that's smell. Now we're going to talk about taste. Okay, so we're going to draw, here's our tongue. Whoop, 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 whoop. There's a tongue, anatomically correct tongue, of course. Inside your tongue, you've got these little hills and valleys. So this is your taste bud. And I'm going to take this and blow it up. So we've got this here, there's a little valley, there's a little plateau, and then so on. You've got bunches of these. Right here, one of the things that we have is we have this little, like, bag. And inside this bag, you can have tons of different like taste receptor cells. And so this has a whole bunch of them. And so each of these cells will detect, as you're eating food, they will detect certain things. So for example, if you are, so salty comes from your sodium ion, sour comes from a hydronium ion, not hydrogen, hydrogen ion, not hydronium. Sweet will come from carbohydrates. I'm gonna write carbs. Bitter, this will come from quinones. Not entirely sure why it's called that. Go talk to an organic chemist. And then another taste that we have is umami. And what does this one come from? I will get back to you on that. once I remember. Actually, okay, so I remembered it's glutamate. Okay, so those are our 5 taste senses. Technically there's more, but this is enough for what we care about. So, and you've got different cells inside this little bag that contain each of those receptors that will go to each of these. So, I'm going to draw a taste cell receptor. So we're just gonna draw a box because it's easy this way. And I'm gonna draw, okay, so here we've got, gosh dang it. Okay, I'm gonna have to do quite a bit of splicing. Okay, so we've got this normal taste cell. So inside each taste cell, you've got a bunch of vesicles, and these will release, if I remember correctly, glutamate onto a connected neuron, then goes to the brain. So let's talk about salty. So this has what's called an ENaC channel. Basically what it is, this is a sodium leak channel. So when you eat something salty, you've got a bunch of sodium ions, and so those will flow into the cell and that will depolarize it. That will then cause the opening of a voltage-gated calcium channel, which will come in, connect to the snare proteins on these, release the glutamate onto this, and cause it to send an action potential telling the brain, hey, I'm eating something salty. So that's for salty things. For sour, it's a little different. So there's a couple things that it can do. So there's still this ENaC channel, but when you're eating something sour, hydrogen ions can come through here and depolarize and cause this same influx of calcium. Or what it can do is it can come in and it can bind to some potassium channels. So it closes this channel up, and what that means is potassium can't leave the cell, so you have a depolarization of the cell because you have less positives on the outside separating from the negatives. So that will cause increase of that, which will then cause this voltage calcium channel to open and then release those and tell the brain, hey, I'm eating something sour. Okay. For sweet, bitter, and umami, they don't have their own specific channel that they're let through, probably because the molecules that signal to the brain that, hey, I'm tasting these things are so large, not little ions, stuff that can't easily diffuse through the membrane. And so each of these has a G protein membrane, which, if I remember correctly, is IP3 activated. Let's double check. Yeah, I was right. Okay. So each of these are IP3 activated. So this increases IP3. And so within here, we've got the smooth endoplasmic reticulum, which has a high concentration of calcium. So when IP3 increases, it causes this to release calcium into the cell, which then releases these vesicles, causes the neuron to depolarize and send a signal off to the brain that says, hey, I'm eating something sweet, bitter, or umami, depending on what kind of receptor this is, if it will attach to those carbohydrates those quinones, or glutamate. And that's taste. Okay, now we're going to talk about sound. This is a super fun one. I'm going to draw an anatomically correct ear. It is not a kidney. There's a little bump that we all have. Okay. That's the outer ear. Don't really care about that. Okay, this is an amazing drawing. I used to be way better. Okay, so we've got this section. And then we've got, now let's use green. And then you've got that. Okay. So just some of the anatomy of the ear. So this is the outer ear. This is what's called your auricle or pinna. That's just the flappy ear thing you have. This is your external auditory meatus, or this is your auditory canal. This right here is what's called your eardrum or your tympanic membrane. When you have air pressure, I'm just going to draw as fun little lines. They will cause this to vibrate back and forth, which will cause these. So these are your little tiny bones inside the middle ear, and these are called ossicles. This is the malleus, incus, and stapes. So what this allows your body to do is it allows it to amplify the pressure, because it has to convert it into this, which is called your cochlea, which is filled with fluid. So because of these, it amplifies that pressure so it can travel through the fluid easier. So membrane pulsates in response to compression of airwaves, which is what sound is. These vibrate, causing the stapes to move in and out of the cochlea, which will cause pressure waves within the cochlea. I'm going to draw this a little better. Okay, there we go. One of the things, so why I did that is, so right here, there's a thing called the round window. And because this is filled with fluid, when the stapes moves in and out, there has to be something that gives so the water can compress and then decompress. So that's what this round window is. It's a little membrane that can pulsate in and out, opposing the movement of the stapes, because you can't change the volume. So what happens is you get pressure from here, causes this moves in and out, and you get pressure. I don't like that there. I'm gonna move that. So it causes pressure in here, which is called your scala vestibuli. So pressure travels up here and then travels back through. And so this green line here is called your basilar membrane. And so if you were to look at it like here and take a cross section, at the front or at the base, it's very skinny. And then as it goes to the apex of the cochlea, it becomes really, really long. And so these length describes the fibers that are within the basilar membrane. And so this is how you get the separation of different frequencies. When you have a high frequency, it causes these thinner membranes to vibrate because it matches their internal frequency. And then lower frequencies will cause these longer, looser fibers to vibrate at that frequency. So now I'm going to take a cross section of this and I'm going to pull it up right here just so it looks, so you can understand a little better. So right here, we've got that scala vestibuli, and then we've got this one, which I don't remember the name. And then here, we've got that intermediate portion, which has this thing, which is called our tectorial membrane, and then we have this, which is called our basilar membrane. And on that membrane, we have fun little I should drop that tectorial membrane a little further. So I shall. Then we have one that's kind of like this. That is not. So this piece right here, that's called your tectorial membrane. And each of these are kind of like little microvilli, right? If I remember, the correct term is stereocilia, are embedded in that tectorial membrane. Inside each of these compartments, they're filled with fluid. In the top and bottom compartments, perilymph. Yeah, both are filled with what's called perilymph. And then inside this compartment, it's called endolymph. That's just because they're made up of different stuff. And also, Latin naming. So here's this, and another important thing is we've got this membrane here, and then we have another membrane here, and then we've got a whole bunch of blood. Okay, so this is kind of like what a cross section would look like. Here, there's something really important that's going on, and I'm going to draw it over here. Yeah. So we've got one membrane and then a second membrane. And so the way that these membranes are set up, I'm going to use black to show that. Black is our sodium ATPase pump. So pulls potassium in, pushes sodium out. And that's the same thing for this one. Purple will be what's called our NKCC. So what that does is it pulls sodium in, pulls potassium in, and then pulls 2 chlorines in. And then on the other side, which I will use green, we have a bunch of potassium leak channels. And so if you're paying close attention, you can already start to see how potassium is moved into here, and then potassium will move into here by concentration gradients. And then we have the exact same thing on this side. And right here is that endolymph. So let's draw those other channels. Here's our potassium leak channel. Here's our NKCC channel. So we end up getting potassium moves into this membrane area, moves into here, and then moves into the endolymph. So we end up getting a really, really high concentration of potassium inside this endolymph. Okay, I'm now going to do a blow up of these. I can draw it here. Perfect. Okay. Perfect. Okay. So the way, then I should draw those. I'm also going to draw those are neurons going to your brain telling you, hey, I'm listening to something, or I'm hearing something. Okay, so I will add that here. Fun fact, as with most of our sensory organs, well, specialized sensory cells filled with vesicles will eventually cause us to depolarize, tell brain something interesting is happening. And so this is sitting, I'm going to erase this. So this sits, we're looking at this one right here. So this is sitting inside that tectorial membrane. And so inside the tectorial membrane, you have a high concentration of potassium per liter. So we're gonna write potassium. Inside here, we also have a high amount of potassium. It's about the same per liter of potassium. That's what that says. I'm gonna move that and make that a more significant K. All right. So we have a high concentration here, and we have potassium leak channels. And so that ends up causing a buildup of negative charges on, the inside. Negative charges on the inside, positive charges on the outside, and then same thing for inside here. And so if you can start to appreciate this, you can recognize that there's a membrane charge here from this endolymph and inside the tectorial membrane and inside the cell and then outside. So what happens is when you have some kind of vibration that comes through from the perilymph, and this membrane here will vibrate, and it will cause the endolymph to vibrate as well at whatever frequency of thing that you're listening to. And it will cause these to vibrate. Each of these little tiny stereocilia have a really interesting chain-like protein where normally they're closed, and so nothing can flow through here. But when they vibrate, it will cause the villi to bend and open up these channels. And so what happens is when these open, potassium will rush in, because in here it is negatively charged. And so this is positive ion. So it's going to come in here, even though it doesn't really do much for the concentration gradient. But electrically, it'll flow in here, and it will start to depolarize this, which will activate, I don't know if it's activate or if we just have some leak channels. I think it will activate some channels here, voltage-gated potassium channels, and potassium will leave the cell here, and it will start to change, and it will start to depolarize both here and here, which will allow calcium to run in and open these vesicles and send an action potential to the brain. Yeah. And that's the ear. Really, really interesting. One of the more important things to recognize is, so you've got these set of those ones that are embedded into this membrane that kind of wiggle up and down with it as the endolymph deals with those pressure changes. But you have another hair cell, which if I remember correctly, these are your outer hair cells and these are your inner hair cells. And so your inner hair cell just sends information to the brain. Your outer hair cells receive and send information from the brain. And so you can kind of cause these to not depolarize as much, which will prevent the brain from receiving more information than it needs to. Kind of like how you can fall asleep while listening to something, say a movie. Your brain starts to be like, this is becoming less important. We're gonna stop depolarizing these cells, so I stop getting really, really annoying information that's useless at this point. And so that is one of the feedback loops that your brain has for hearing. I think that's everything. So we went over muscle spindles, Golgi tendon organs. We described how smell works. We described how taste works, especially with salty and sour and their specific cation channels, and then sweet, bitter, and umami through their G protein pathway. And then we talked about sound. We talked about the anatomy of the inner ear and quite how the ossicles work to advance pressure waves through the cochlea. We talked about the basilar membrane. High frequency things will be heard more at the base, and then lower frequencies will travel further and vibrate those fibers at the apex of the cochlea, the basilar membrane. And then this is how your action potentials transferred. Shaking here, these open, potassium rushes in here, and then potassium leaves here, depolarizing the membrane, calcium will come in, and then send a neurotransmitter here, depolarizing that neuron, sending signal to the brain saying, I'm hearing something.